Okay. Hello, everyone. My name is Lina Morales. I'm a fourth year neuroscience major here at UCR, and I will be presenting my emerging research on the embryonic development of GnRH neurons in a mouse model of Fragile X syndrome. So Fragile X syndrome is a genetic disorder caused by mutations in the Fragile X mental retardation gene. This often results in various developmental disorders, such as autism spectrum disorder, intellectual disabilities, Fragile X-related tremor or ataxia, as well as reproductive disorders, such as macroorchidism in males, which is enlarged testes, and premature ovarian failure in females, which is a condition where women experience early menopause before 40 years of age. Since Fragile X syndrome is highly associated with autism and intellectual disabilities, the effects of FMR1 mutations on regions like the hippocampus and the cortex is progressively studied. However, the effects of these mutations on the hypothalamus is not studied. So the hypothalamus is an essential region for regulating reproduction. Normally, hypothalamic neurons secrete GnRH, which act on the anterior pituitary via the hypophysioportal system. GnRH is secreted in a pulsatile manner, and that's what regulates the secretion of LH and FSH from the anterior pituitary. Since FMR1 mutations are resulting in the development of reproductive disorders, it's essential that we analyze the underlying mechanisms causing these issues. So our lab specifically uses an FMR1 knockout model. This knockout model mimics the lack of Fragile X mental retardation protein, which is similarly seen in individuals with Fragile X syndrome. Specifically, we are using this model to analyze how the lack of FMRP is affecting reproduction. So to begin this analysis, a prior fertility study done by Pedro Villa, a graduate student of the COS lab, paired 8-week-old knockout and control female mice with control males and allowed them to reproduce until reproductive function stopped. It was found that FMR1 knockout females stopped reproducing earlier compared to wild type. Additionally, pups per litter were analyzed, and it was found that FMR1 knockout mice were giving birth to larger litter sizes compared to wild type. Since they saw this increase in litter size, the COS lab decided to count corpora lutea number within the ovaries because it's a structure that indicates ovulation has occurred. It was found that FMR1 knockout mice had increased corpora lutea numbers compared to wild type, which could explain the larger litter sizes seen in the prior study. Additionally, similarly to what occurs in individuals with FMR1 mutation, these female mice are experiencing some form of early cessation of reproductive function. So that data in correlation with the fertility study prompted the COS lab to look into the hormones responsible for ovulation and follicular growth. They specifically analyzed Fragile X syndrome, FSH and LH. FSH is essential in regulating follicular growth, and LH is essential in stimulating ovulation. It was found that in knockout mice, both FSH and LH were elevated in knockout mice compared to wild type. Since GnRH neuron pulsatility is what regulates the secretion of these gonadotropins, these elevated levels are indicating that FMR1 mutations are causing altered GnRH neuron activity. So in order to get a better insight into how FMR1 mutations are affecting GnRH neuron function and how that relates to hormone secretion, Pedro and I performed an LH pulsatile analysis because with every pulse of GnRH, there's a direct and matching pulse of LH. So in order to do this analysis, we staged mice, and once they were in diestrus, we collected 10 microliters of tail tip blood for every 8 minutes for a span of 3 hours. When comparing the LH pulses of knockout and wild type mice, we found that the knockout mice showed increased LH pulse frequency compared to wild type. And that gives us insight into GnRH neuron activity, suggesting that FMR1 mutation is increasing GnRH neuron pulsatile activity, which could be increasing LH LH pulsatility, which could explain the elevated LH levels and increased corpora lutea seen in the prior studies. So since we are observing this altered GnRH neuron activity, and since Fragile X syndrome is a developmental disorder, in a future study, we want to investigate how GnRH neuron development and is affected by FMR1 mutations. So normally around embryonic age 10 to 11, GnRH neurons begin development in the nasal placode. Then they migrate towards the nasal forebrain junction. And around embryonic age E15.5, GnRH migration rates decrease and the neurons reach their final destination in the hypothalamus. So my specific question for this future study is, do FMR1 mutations cause irregularities in GnRH neuron development? I will analyze the development of GnRH neurons in 5 pairs of wild type and FMR1 knockout mice at embryonic age E15.5 because it's a critical stage of GnRH neuron migration. I will then quantify the differences in GnRH neuron number as well as GnRH neuron migration patterns in order to determine if FMR1 mutations are causing differences in neuronal development. So in order to do this, we will be fixing E15.5 embryos in 4% paraformaldehyde on ice for an hour. Then we will be freezing these embryo brains in isopentane on dry ice. Then I will be sectioning these brains using the cryostat. The sections will be sagittal sections at 30 microns. And in order to visualize GnRH neuronal cell bodies within these brains, I will be using immunohistochemistry staining. So these are some pictures for my optimization trial. Right here, you can see this would be the mice's brain, the mice's skull. Right here would be the jaw and the tongue. Here you can see the nasal folds. And if we zoom into this box right here, you can see stained GnRH neuronal cell bodies, which seem to be migrating near the nasal forebrain junction, which is seen right here. So again, I'll be using this staining technique in order to quantify the differences in GnRH neuronal number and migration patterns to see if FMR1 mutations are affecting the development of GnRH neurons. I would like to give a special thanks to all the members of the COS Lab for all their guidance and help they've given me throughout my research experience. Also, I would like to thank all of you for being here and for your attention. I really appreciate it. Thank you. Any questions?
Thank you very much, Lina. Great job. So we now have plenty of time for questions. If anybody has any questions for Lina.
I have a question. I need to learn how to raise my hand on this thing. So you did talk about this a little bit, but can you tell us a bit more about kind of why your specific research is interesting or exciting? Like, I know how it connects to the research that's already been done in your lab, but why are you focusing on this specifically?
So specifically, we're looking at this because, like I said before, the Fragile X syndrome is highly associated with autism and intellectual disability. So there's a lot of research on how it's affecting the hippocampus and the cortex. However, there's not a lot of research on the hypothalamus. And since the hypothalamus is an essential region for reproduction, and we're seeing these reproductive disorders develop because of FMR1 mutations, we want to focus on how it's affecting the hypothalamus so we can get more insight onto why these reproductive disorders are occurring in individuals with FMR1 mutation.
Thank you.
Great. Any other questions? Oh, Byron has a question.
Yeah, good morning. Wonderful presentation. I'm asking a question because I unfortunately couldn't get into the beginning of your seminar. But how was the fMRI knockout done? Was it knocked out all over the body, all over the brain in a specific cell type?
From what I know, it's a full knockout. So
Okay.
There's the lack of expression of FMRP, which is a Fragile X mental retardation protein. So it's a complete knockout.
Thank you.
Excellent. Any other questions for Lina? Well, please join me in thanking Lina again for a great presentation. We'll now move back to the beginning of our lineup to Arya Sharafat. Arya, I'll introduce you again.